Hello, everyone. Welcome to this webinar on bacterial cell wall as a living biopolymer, brought to you by the Protein Society. My name is Liz Meyering. I'm the president of the Protein Society, and I'm delighted to introduce our webinar today on this very important topic in protein science. Before turning the proceedings over to our organizer for today's webinar, Shahriar Mobashery, I'm going to make a few brief announcements. First of all, if you have suggestions for webinar topics or would like to organize a webinar, please contact us. You can see information about this on our society webpage for webinars. I also want to give you a heads up that our next webinar is on protein science in South America, and this webinar will be on March 4th. So there'll be more information coming to you. And I'd also like to invite you to join us for our annual symposium in San Francisco this year, June 26th to 29th. We have a very exciting program. We have a lot of travel awards for young scientists. And again, you can find more information about the symposium on our society website. I also want to highlight our flourishing journal, of Protein Science, which is led by our fantastic editorial team, led by John Currian, and it publishes a wide range of protein science, and I encourage all of you to consider publishing your latest research here as well. I also want to highlight that we have Best Paper Award winners each year, where a junior author is designated as the award winner and is invited to give a talk at the annual symposium, along with other benefits that go with the award. And now to our main event. Again, I want to thank Shahriar Mobashery for bringing us together to hear about the exciting research that's being done by these researchers here on this topic that is of tremendous fundamental and medical importance. A bit of background on Shahriar is he obtained his BSc in chemistry as well as biological sciences from the University of Southern California, then his PhD in chemistry at the University of Chicago, followed by postdoctoral studies at the Rockefeller University. He then started as a faculty member at Wayne State University and subsequently has held the position of Navari Family Professor in Life Sciences at the University of Notre Dame. Among his prestigious recognitions are being Fellow of the American Association for the Advancement of Science and Fellow of Science Without Borders. As well, he received the Kaiser Award from the Protein Society in recognition of highly significant contribution to the application of chemistry in the study of proteins. So I'm not going to try to tell you all of his recognitions, but his research program integrates these fields of chemistry and biology with computation, biochemistry, molecular biology, and organic synthesis of medically important molecules. And this multidisciplinary approach has led to scientific and medical advances in critically important clinical problems, such as antibiotic resistance and diseases of the extracellular matrix. So I'm very much looking forward to hearing his presentation today. I'd also like to thank our other speakers, Juan Hermoso and David Roper, for sharing your exciting research with us today as well. And I also want to thank our staff at the Protein Society, Raluca Kadar and Shannon Cunniff, for all your help with the webinars. So before I turn this over to Shahriar, just a few logistical notes. So the way that this webinar is organized is each speaker presents for approximately 30 minutes, including a roughly 5-minute question period. And following all three talks, so we'll have a few questions after each talk, and after all three talks, we'll have further questions in a discussion format among our speakers. So please, everyone, take a moment now to find your Q&A tab in Zoom. And you can type your questions there at any time. Only the organizers and speakers will be able to see the questions. And then Shahriar will be reading out questions, which may not all be answered in real time, but speakers may also type answers to questions in the chat. And then we'll return to some questions at the end as well. And these questions that come from the audience enrich our webinar so much. So I really encourage you to share your thoughts as well. And with that, I will turn it over to Shahriar. There you are. Hello.
Thank you, Liz, for the very generous introduction. I'm delighted to be here in organizing this webinar. I thank Protein Society for agreeing to host us. It has been a wonderful experience. As you heard, we are talking about cell wall in bacteria, and what I would like to point out is that cell wall is a biopolymer of enormous dimensions. It is, of course, cross-linked. So what that entails is that it is a single molecule that encases the entire bacterium. So to the right, you're seeing here atomic force microscopy images of E. coli that we reported a number of years ago. And schematically to the right, what you see is an outer membrane, and I didn't mean to do that, an outer membrane and inner membrane, and between the two, you have the cross-linked polymer cell wall. Arguably, cell wall is one of the last important biopolymers for which the three-dimensional structure is not known, and there are at least several dozen enzymes that do various things to put the cell wall together and regulate its function. And as we'll learn, there is also recycling. With that said, I'm quite pleased to share the medium with two distinguished speakers, David Droper from the University of Warwick in the UK, who will talk to us about assembly of cell wall. Juan Hermoso from Consejo Superior de Investigaciones Científicas, who will talk to us about regulation of the processes that govern cell wall. And at the end, I will have the pleasure of talking to you about the repair mechanisms for bacterial cell wall, as well as its recycling. So you're going to see a full range of things that happen with bacteria. With that said, I turn the table to David, and we look forward to hearing from you, David. Let me see. Okay.
Okay, wonderful. Thank you so much. Let me just make sure we can see this. If someone could tell me that we're on full screen for the audience, that would be wonderful. So thank you very much to the Society and to Shahriar and Juan for the opportunity to speak here today. It's a great honor. My apologies if my voice is a bit croaky. Both Juan and I were in the sunshine and warmth of South Africa last week, and returning to a wet and cold Heathrow on Sunday morning was a little bit of a shock to the system. But anyway, we'll try our best. So my job is to talk to you about some of the basics of bacterial cell walls and how some of the enzymes involved are what they do, and a perspective on what I think may be an avenue to new antibiotic compounds. So, oops. So there are lots of different antibiotic targets in bacteria, which are on this slide here. We got metabolic inhibitors affecting all sorts of different aspects of cellular metabolism, but arguably protein synthesis in the ribosome is a major target for antibiotics, some of which you can see listed here on the screen. Again, nucleic acid biosynthesis, and particularly the DNA gyrase enzymes and disruption of that process is efficacious for antibiotics. But arguably, the greatest target for the antibiotics that we have is the bacterial cell wall, and of course the beta-lactams, which have saved lives since the 1940s, are one of the mainstays of antibiotic chemotherapy. So, what is it about the cell wall that's important? Well, the cell wall, in particular peptidoglycan, gives cells shape. And as Kevin Young eloquently put it, to be brutally honest, few people care about bacterial cell shapes, which is a shame because the bacteria seem to care very much. And in fact, they put a large amount of their energy and metabolism into this process. And it underlies the fact that the cell wall is absolutely vital to bacterial metabolism and growth and division. And as you can see from, we have all sorts of different cell shapes, from the spiral spirochetes to the typical rod-shaped bacterium that we see in the middle, and even to very rounded bacteria like Staph aureus. So what is peptidoglycan? It's a polymer. It's a polymer that exists outside the cell membrane and in the so-called gram-positive organisms like Staph aureus and Streptococcus pneumoniae. That's a very thick layer that exists above the cell membrane. And in the so-called gram-negative bacteria like E. coli and Pseudomonas, that's quite a thin layer, which is sandwiched between the cytoplasmic membrane and the outer membrane. But in either case, the peptidoglycan layer is integral and important for cell shape. So what is it? It's an alternating sugar polymer of N-acetylmuronic acid and N-acetylglucosamine. And the sugar glycan strands are linked together by peptide bridges, which either contain meso-DAP at position 3 in the peptide sequence that you can see in front of you, or lysine at position 3. And interestingly, these peptides contain, in some places, D amino acids. Okay, so we envisage that this peptidoglycan layer is made up of spirals, if you like, of these glycan strands around the cylindrical circumference of the bacteria, linked together by the peptide stems, and there are a group of enzymes, which we'll come to in a minute, the PBPs, the penicillin binding proteins, which mediate this cross-linking process between adjacent peptide stems. And this involves the very terminal D-alanines in this pentapeptide stem, and penicillins, beta-lactam compounds, essentially are mimetics for that D-alanine terminus. And weakening the cell wall through the action of beta-lactams to inhibit the cross-linking process leads to weakening the cell wall, osmotic lysis, and so on, and cell death. And what does that look like? So here we have two, oops, beg your pardon. So here we have two examples of failure in cell division. Sorry, failure in cell division and the activity of some of these PVPs. So on the left-hand side of the screen, you see a failure in the PVPs responsible for cell division, which leads to a bulging and eventual lysis. And on the right-hand side of the screen, you see what happens when you inhibit the enzymes responsible for cell elongation. Basically, you change shape. You go from rod shape to round. So how do you make a cell wall, or how do you make peptidoglycan? Well, there's a cytoplasmic phase of biosynthesis in which UDP-linked sugars are converted from glucanac to mernac. The pentapeptide chain is added to make the UDP-mernac pentapeptide soluble precursor, sometimes known as the PARC nucleotide. That is then linked to an undecaprenol phosphate carrier lipid at the inner surface of the cell membrane by MRAY to form lipid 1. You then have the addition of a second sugar, the UDP glucanac, by MERG to form the all-important molecule lipid 2, which is flipped across the membrane and then is accessible by the PVP enzymes for the first reaction to form a glycan strand, glycosylation, and then for a subsequent reaction, again catalyzed by the PVPs, which is transpeptidation, this cross-linking activity, which is inhibited by beta-lactams. So this lipid 2 molecule is the all-important building block of the cell wall. It contains this NAG-NAM disaccharide core with a pentapeptide stem, which varies in composition, whether you're gram-positive or gram-negative, and then the C55 carrier lipid undecaprenol pyrophosphate. And the literature is absolutely full of studies on the cross-linking reaction, which is inhibited by the beta-lactams. But my focus in my research is at the earlier stage, that glycoside transferase step, which I think is the next great avenue for antibiotic discovery. And I'll tell you a little bit more about that as we go along. So, firstly, what are the PBPs? Well, the glib answer is the worst named enzymes in the world, because PBP doesn't tell you about their enzymatic activity, just about the antibiotics that they bind. So penicillin binding proteins are actually peptidoglycan synthase or peptidoglycan biosynthesis enzymes, and they come in 3 classes, only 2 of which we will consider in this talk today. The so-called class A's are high molecular weight, and we're talking in the range of 80, 90, sometimes 100,000 molecular weight, and they're bifunctional. They have both the glycoside transferase activity and the peptide cross-linking activity, the transpeptidase. And typical examples are E. coli 1A and 1B. And then we have class B enzymes. These are monofunctional. These can do the transpeptidation reaction anyway. And some good examples are E. coli PBP2, which is involved in elongation, and PBP3, which is involved in cell division. So in this little movie, we have a really nice visualization of what's actually going on. So this is E. coli PBP1B from the crystal structure from 2009. We have modeled in the active site a version of lipid 4, so the first reaction product, and then along from the right-hand side, we have lipid 2 coming along. And then what we start to see is the formation of this long glycan strand in the orange color with the green peptide sticking off the side, which as it gets longer and longer, is able to then make a connection with the transpeptidase site. So we have in this depiction, we have a red glycoside transferase domain, a blue transpeptidase domain responsible for the cross-linking, and then in this particular example, the UB2H domain is involved in regulation. At the heart of this glycoside transferase process is the active site. Excuse me, and the formation of this glycan strand is itself a processive mechanistic process. We have a so-called donor site, which is where the growing glycan strand is formed. And then we have an acceptor site where the incoming lipid 2 molecule can bind, and that enables then the beta-1,4 glycosidic linkage to be made at the active site of the enzyme. Everything moves essentially from right to left, with the release of an decapropenyl pyrophosphate molecule at the end. So we go round and round and round this circle to make a long glycan strand. There is an all-important inhibitor of the process called monomycin, a natural product with no natural resistance that is known, which binds with nanomolar potency to the donor site of the active cell. So how can we visualize this? Well, we can make lipid 2, one of a few groups in the world that can make lipid 2. We make a fluorescent form with danzol off the peptide stem. We can polymerize that with the enzyme, run the reaction products down and polyacrylamide gel, and visualize the products. And this is the kind of thing that you see on the right-hand side. You've got a nice long polymer of lipid 2, so you can count lipid 2, 4, 6, 8, 10, etc. as we go up the strand length. So we have a nice way of visualizing the activity of these enzymes. Now, making inhibitors for this process is tricky, because the lipid 2 molecule itself is lipidic. It's a difficult compound to form mimetics of. Many have tried. There's a lovely review article that you see down here for 2016 with lots of different compounds. But we started to engage in this several years ago with colleagues from the University of Queensland, who came up with a couple of compounds, ACL 20215 and 20964, which were designed to be lipid 2 mimetics. They certainly bound and inhibited a glycoside transferase enzyme from Staphylococcus in vivo, and actually they work in a mouse mastis model of infection as well. So that's really important. They're efficacious at the site of infection if injected directly there. Unfortunately, not so good in the serum, where they get bound by serum plasma proteins. But interestingly, these compounds have a very low level of resistance development, suggesting that they're multi-targeting, which is an important facet of antimicrobial design. We published this back in 2015, and we've been doing some work subsequently from there to try and really figure out what's going on. So here are some of those studies, which is not published at the moment, but I will tell you anyway. So we use our gel-based system to look at the activity. We've found IC50s for these two compounds, 201-215, 209-64. But what we also noticed was that the chain length over time with these compounds was being reduced, shortened. And that gave us a thought and a clue about what might be going on. So we do a little bit of crystallography in my lab. So we grew some crystals, but unfortunately only in the presence of monomycin, that inhibitory compound, but also in the presence of ACL20215. And what we found was that these compounds are actually binding not at the active site, but adjacent to it, in which we think is the tunnel through which the growing glycan strand emerges. So we now have this conceptual model, which we're still pursuing, of what's happening here. And we think what they're doing is they're binding in the glycoside transferase exit site, so not in the active site directly. And if you like, it's like putting pushing a cork into a bottle of wine, restricting the flow. And if you make short glycan chains, that is not compatible with life, and that's why they're probably efficacious in vivo. However, so that's a story about the class A enzymes, which 10 years ago we thought were the most important factors in cell wall biosynthesis, because they did both this glycoside transferase, lipid 2 polymerization step, and also the subsequent transpeptidation. But if you look at the affinity profiles of the clinically used beta-lactams, you will find it's not the class A's that are important. It's actually the class B's. The PBP2 or the PBP3, which I talked about before, they have much higher affinity for the clinically used antibiotics. And that's because we've discovered recently that these class B enzymes form complexes with so-called shape, elongation, division, and sporulation proteins, SEDS proteins. And this intimate complex does the two reactions. So the SED is the glycoside transferase, and the class PBP provides the accompanying transpeptidase activity. And these complexes are important for both the rod system that provides cell shape. So in E. coli, for example, you have rod A and PBP2, which is the central enzymatic engine that drives cell shape construction. And then you have FDSW, PBP3, sometimes known as FDSI, at the heart of division, and again assisted by other proteins to give you peptidoglycan biosynthetic activity at those two sites. So these are the engines which drive cell wall biosynthesis and are assisted by the class A enzymes. And this is where Shahriar talked about science. One of Shahriar's multifaceted skills is he's involved with an organization called Science Without Borders. And certainly that helped with me back in 2018 when I visited a good friend of mine, Jonathan Dawkin, at Columbia University. And he took me to see his colleague, Filippo Mancia, over in a different department at Columbia. And they showed me something which I found absolutely fascinating and has led us to where we are today. So Filippo had in his group a group of researchers who were looking at the rod A PBP2 complex of E. coli, and they showed me these electron micrographs. And interestingly, you can see here the PBP from these class averages sticking out the side, and the rod A enzyme is encased within this membrane scaffold protein, which looks like this thing here. So you can see here the beginnings of a cryo-EM structure experiment, and I was lucky enough to go over to Columbia in 20 and 21 on sabbatical to pursue this structural story with them. So they have a fusion, or we have a fusion of rod A and PBP2, which we can express and purify. That is a membrane protein, integral membrane protein. So we use the membrane scaffold protein technology to extract that from membranes in a detergent-free context. And we were able to initially determine a 5 angstrom structure and then get us down to about around about 3 angstrom structure, which you see here. So this is fascinating. What we have is the single transmembrane helix of the PBP inserting with the 10 transmembrane helices of rod A, and that insertion causes a shape change in the rod A, which activates it as a glycoside transferase. Here we see two different views, and I'll draw your attention to this helix turn helix motif here in a second or two longer. So this was the basis for our cryo-EM structure. This is the engine at the heart of the elongosome. There are a group of other proteins, MRE, B, C, D, rod Z, PP1A, which associate with this complex and modulate its activity and its ability to deposit new peptidoglycan for cell elongation. So here's some work that we have published and we are still pursuing in and around all of these SEDS proteins. I'll draw your attention to 3 residues. There's the tryptophan at position 102 in the E. coli structure, an arginine at 210, and an aspartate residue at 262, which are important. So the W102 residue is unfortunately not in our density. It's on a mobile helix. Arginine 210 is on this helix turn helix motif that I pointed out a moment or two ago. And the aspartate 262 is in a loop region adjacent to what we think is the active site of rod A, and was previously identified by a group of researchers at Harvard who did the comparable thermophilus structure of rod A and rod A PMP2. So using our gel-based assay system, we can assay mutants. So for example, the aspartate 262 to alanine mutant completely knocks out the activity of this protein, and we think that's the all-important catalytic residue. But it's more than just the catalysis. We've got a whole range of different arginines in and around the active site, which we think are holding the 2 lipid 2s at the donor and acceptor site of the enzyme at the right time to enable catalysis. W102, if we substitute that with alanine, we kill the enzyme stone dead. If we put there phenylalanine, we restore activity. What we think that's doing is guiding the glycan strand out to the active site. And the arginine that I pointed out at 210, that is involved in the helix turn helix motif, which binds the decaprenyl pyrophosphate, which is released from the reaction. And that motif with the arginine is actually found in a range of other enzymes, which also handle, be they transferases or polymerases, and decaprenyl phosphate carriolipids, some examples of which you can see here. Using the genius of one of my colleagues, Phil Stansfield at Warwick, who was a computational simulations expert, we were able to model both the 2 cavities for the lipid 2 binding site and exploring many of the residues which are involved there. We come up with a mechanism, which is in our paper, and for the sake of time, I won't go into that now, but we think we now have a handle on what's going on. And then we have this beautiful little model or simulation of the glycoside transferase activity. We see movements of the rod A protein as it polymerizes and grows a glycan strand, which snakes out of the active site towards the transpeptinase site. Along at the same time, a group at Cambridge, headed by Jan Löwe, came up with a comparable structure for the divisome specific complex, FDSW, FDSI, with QLB bound as well. And they essentially see a complex of the core complex of the SEDS PBP, which is pretty much identical in many ways to ours. So this is a conserved structural architecture of these all-important enzymes. Interestingly, if you put the sequence of these proteins into AlphaFold, you get not the folded over experimental structure, but an extended structure, suggesting that the enzyme undergoes a conformational change, and that's exactly what we think happens. So we think there's a conformational change between the down ground state structure that we see in our experimental structures and the up state, which is probably the representative more of the real active form of the protein where it makes peptidoglycan. That's led us into a very happy funding situation in the UK where we've now got a collection of researchers and plenty of money for 5 years to pursue that, and that's exactly what we're doing. So that's really the end of my whistle-top tour of peptide-like and biosynthesis. There's a meeting called the Great Wall Symposium, which we've been convening for the past decade or so, which tells you a lot more. So if you're interested in cell wall biosynthesis, I encourage you to attend. So thank you to all the people who have helped me in these explorations, particularly my colleagues, Jonathan and Filippo at Columbia University, and the wonderful opportunity to visit that university at the end of the COVID pandemic. And of course, to all my colleagues here at Warwick. So with that, I will stop and stop sharing my screen. Thank you very much.
Thank you, David. I invite everyone to send in their questions. Presently, there are no questions, so I will ask a couple.
Mm-hmm.
Regarding that prediction by AlphaFold for the upright arrangement for PVP, of course, AlphaFold needs to have precedent to assemble the structure that it proposes. Are there examples that mimic that upright position that they see, at least in AlphaFold?
I'm probably the wrong person to ask this of. Phil, for my colleague Phil Stansfield is a better person to ask. Certainly of the SEDS PBP complexes, the only experimental data are the E. coli rod A PBP2, the Pseudomonas FDSW PBP3, and the Thermatoga rod A PBP2, which are in the database. However, there are many structures of individual PBPs available in the database. So whether they have influenced that prediction or not, I presume the answer is yes. That may be the case. But it actually makes a lot of sense because the experimental, as it were, L-shaped structure we see is not going to be competent to form the polymer. We need that glycan strand, which is being made at the cell surface, to extend all the way up to the peptide-glycan layer, when there is definitely a separation between the two. So it makes total sense that there is a conformational change. And what's more, in the cryo-electron structures that we have, we definitely see different class averages where we see different arrangements of the PVP relative, as it were, to the SEDS protein. So it looks likely, and there's experimental data to support that motion as well.
Very good.
Juan.
I have, if I may, Juan has a question. Go ahead, Juan.
Okay, very nice presentation, David. So I wonder, you have presented a really very nice mechanistic implications for the glycosyl transferase activities in different kinds of proteins, PVPs and SEDS proteins. So I wonder if the mechanism is exactly the same in both cases. I mean, is nature able to really find two different solutions for a similar mechanism? This is my first question. And the second one is there any kind of inhibitor? You have presented moemomycin for PVPs, but is there any other kind of inhibitor for SEDS proteins?
Okay, so do the class A's and the SEDS proteins have a similar mechanism or something different? I think was the first question. And the answer is in part. Some of the residues used for binding and catalysis are similar. I have to say that in retrospect, I'm not sure that we fully understand what's going on at the glycosyl transferase site, the class A enzymes. We don't have good crystal structures of the class A enzymes with a substrate or a substrate mimetic. There are lots of structures with monomycin, but monomycin is a product mimetic, not a substrate mimetic. So yes, some similarities, quite a few differences. We have recently been doing some studies with monomycin against the SEDS proteins, which we initially thought was not an inhibitor. I think this answers your second question. But at high concentrations, we do see some level of inhibition. And recently, about a year ago, Suzanne Walker's group at Harvard published a paper which they then did a correction to of a molecule which they discovered in a screen which seems to inhibit the SEDS proteins. And we're looking at that one as well. So the jury's out. I have great hopes that this is going to be an avenue for new antimicrobial discovery. And hopefully these kind of compounds will not only just inhibit one enzyme, but they will inhibit both the SEDS proteins involved in elongation and division. And if we hit both things at the same time, how will the bacteria get over that? It's not going to be through mutational resistance.
Okay.
I hope.
David, one question that has come up is that what are similarities between gram positives and gram negatives with respect to the sort of machinery that you have talked about?
It's all pretty similar. So the nomenclature of PBPs differs between organisms, which is one of the problems in the nomenclature of those enzymes. But the basics of class A PBPs in all organisms, be they gram negatives or gram positives, is pretty similar. We have different domains which allow regulation. There are subtle nuances in antibiotic binding, which you published on, Shahriar. So I'll direct that question back to yourself. And with the SEDS proteins, the logo plot that I put up shows you that there is very high sequence similarity and conservation of the residues, which are important at the heart of these enzymes. What's more, some of those residues which are conserved are not actually concerning the mechanism of the enzyme itself, but in the protein-protein interactions that these proteins need to make in order to form functional complexes for both cell division and elongation.
Thank you, David. There are several other questions, but we need to move on, and we'll come back to additional questions at the very end if time permits. Thank you. So I will close this section by making a comment, David, that the rate of the frequency of mutation that you're observing is the background mutation rate of bacteria. 10 to the minus 9 to 10 to the minus 10. So that's pretty good. Maybe some passage experiments with the compounds would be useful to see how readily resistance emerges. So with that said, I'm going to stop, and we'll move on to Juan Hermoso from CSIC in Madrid, Spain.
Okay. Let me share the screen. I hope you can see my screen. So first of all, I would like to thank Protein Society for kindly invited me, and also Shahriar for sharing this presentation. So today I will focus my talk on the regulation between the cell wall degrading enzymes in bacterial cell division. So as nicely explained by David, so the bacterial cell wall is composed by membranes. So in the gram-positive bacteria, we have an inner membrane, while in gram-negative bacteria, we have the inner and outer membrane, and in between, there is the peptidoglycan. So the cell wall is a gigantic macromolecule that defines the shape of the bacterium and enables the bacterium to resist the high lysis pressure that is inside the bacteria. So in that sense, all the proteins that are involved in cell wall remodeling are essential targets for design of new antibiotics. Peptidoglycan is composed by disaccharide, is composed by N-acetylmuramic acid, N-acetylglucosamine, as explained by David before. So there is attached to the NAM units, there is the peptide stems that are cross-linked during the glycosyl transferase and transpeptidation process in order to create the framework. So this peptidoglycan is involved in many critical events in bacterial life, such as cell division, antibiotic resistance, cell wall recycling, cell growth, flagella and pill insertion, human interaction, sporulation, and of course, morphogenesis. So as you can see, this peptidoglycan is at the core of many essential processes in bacteria. So as you can imagine, so there is two different machineries, the biosynthetic machinery and the lytic machinery. The first one has been explained by David, in which we have glycosyl transferase activities that are making the polymerization of the glycan chains, and also the transpeptidations that are making the cross-link in the peptide stems. But together with these biosynthetic machineries, there is also the lytic machineries that are specific enzymes that are able to cleave specific places on the peptidoglycan framework. So there are some glucosaminidases or exolytic transglycosylases that are specifically cleaving the glycan chains. And also there are some proteins such as peptidoglycan amidases, endopeptidases, or carboxypeptidases that are cleaving in the peptide stem of the bacterial cell wall. So as you can imagine, so the different synthetic and lytic machinery must be carefully regulated to warrant bacterial survival. So in our group, we are working all different aspects of microbial structural biology. First of all, by designing new kind of antibiotics or new kind of therapies, such as antibiotics. We are also trying to understand, to uncover the molecular basis of antibiotic resistant mechanisms in different multi-drug resistant pathogens. And for many years, we have been involved in the study of how bacteria infect us, and also how is our own response. And in between, we are also studying fundamental processes that are concerned the bacterial cell wall. In that case, I will focus just on regulation of peptidoglycan hydrolysis in cell division. So the main machinery for cell division is the so-called divisome. Divisome is a machinery that is composed by dozens of different molecules that combines the biosynthesis and lytic machineries, and that is more or less preserved between gram-positive and gram-negative bacteria. This divisome contains some specific domains, such as spore domains, that are involved in specific recognition of septal peptidoglycan. But also there is a very important and crucial machinery is regulated by the membrane complex FTSC, FTS6, and that controls the lytic activity of the proteins that are involved in the separation of daughter cells at the final part of the cell division. Together with this, in gram-positive bacteria such as Streptococcus pneumoniae and some relatives, there is other lytic enzymes such as LYT-B that surprisingly has been discovered to be regulated by serine kinases. So in this talk, I will focus on this system in order to provide some insights into the molecular details of this regulation. So concerning the first part of my talk, concerning the specialized domains targeting septal cell wall or lysins, so it has been discovered that there is a specific kind of domain called LYT-M that is very important in regulation of this lytic activity by some kind of protein-protein interactions. Indeed, LYT-M domains are originally presenting lytic activity, but some inactive conformations of these LYT-M domains allow the recognition of other lytic enzymes, such as amidases, and importantly, they are activating these amidases. We have seen recently that LYT-M in Caulobacter crescentus presents a conserved groove that is acting as a docking station for different autolysis. Another important domain is the so-called spore domain that is presenting a specific structure allowing recognition of the denuded septal peptidoglycan. This is the peptidoglycan that is produced at the end of the activity of amidases that are cleaving the peptide stems. So we have seen that through a combination of recognition of main chain interactions and specific basic patches that are located in the binding site, So these spore domains are able to recognize specifically the new receptor peptidoglycan that is only produced during cell division. Okay, so concerning the second part of my talk, the regulation by the FTSE-FTS6 system. So FTS6 regulates peptidoglycan hydrolysis in both gram-positive and gram-negative bacteria. And this system is involved in both cell division or in cell elongation. As you can see in this slide, so there are different components. In the case of E. coli, for instance, on the left part of my slide, you see that FTSE is expected to have ATPase activity, and FTS6 is crossing the membrane in order to support the MC protein, that is some kind of pedestal protein presenting the lead M domain we have talked previously. And then attached to this, there is an amidases that are performing the lytic activity. So in the case of gram-positive bacteria, there is no MC protein, and there is a direct connection between the FTS6 and the lytic protein. So interestingly, all this system, these proteins belong to the family 7 of ABC transporters. But contrary to many other ABC transporters, there is no transport of molecules, but this system performs mechanotransmission from cytosol to periplasm fueled by ATP hydrolysis. So we have started the study of this system some years ago by the structural and functional characterization of essential PCSB protein in pneumococcal division. As you can see here, so the protein, as observed by crystallography, presents a very long coiled coil regions, and together with this coiled coil, there is a alanine-rich linker connecting with the catalytic domain or CHAP domain. Interestingly, in our crystal structure, we have observed that the protein was making a dimer in such a way that this dimer is performed by 3-domain swapping and presenting the active site occluded by the coiled coil of the dimeric partner. But even at low resolution, we have observed that the active site of PCSB is inhibited by the coiled coil. So very likely, this protein requires the activity of the FTS6 system in order to really activate or release the catalytic domain. So we have continued this study by structural and functional characterization of FTSE. We have seen in the crystal structure, we have solved a very nice resolution. That FTSE contains all the elements expected for ATPase, such as the worker A, worker B, or the different components that allow this ATPase activity. But together with these shared elements, so we have seen that FTSE presents unique regions on surface that is showing a strong plasticity, and this plasticity very likely connect with the interaction with the other partner, FTSX. But only recently, in a very nice collaborative effort with the group of Luo Ming and Chris Sam at University of Singapore, we have provided the cryo-EM structure of the complete system, the membrane complex between FTSE and FTSX in Pseudomonas aeruginosa. So in that case, we could really identify the different components, such as membrane, transmembrane segments that are propagating the signal from the cytosol to the periplasmic space. Interestingly, we have seen that in the absence of ATP, so the extracellular domains of FTS6 that are indicated here, so were extremely mobile, while in the presence of ATP, there is a further stabilization of these extracellular elements. More, so we have included MC, the pedestal protein in this complex, and we see that, so we can see how the coiled coil of MC here in magenta is recognized by the extracellular domains of FTS6. But in the absence of ATP, most of the MC protein comprising the lead M domain remains completely disordered. There was no information, no structural information for this domain. But surprisingly, when we add the amidase, so we can see that we have the full supercomplex in which we have FTSC, FTS6, MC, and AMIB, the amidases. So as you can see, this at that time we have a very long structure, really around 300 Armstrong over the membrane, and we have the different components of this activation. So we have ATP hydrolysis that makes this compression of the transmembrane segments in order to push the coiled coil of MC that provides this extension of the coiled coil in order to put the Lytic domain here in red on this 3 Armstrong over the membrane. And finally, this Lytic domain is able to activate the amidase by release of the helix that is blocking the entrance of the substrate to the active site. So this provided the mechanotransmission, the molecular basis of the mechanotransmission in Pseudomonas aeruginosa. So this is the proposed mechanism indeed. And also we have seen that this mechanism is very well preserved in the E. coli bacteria. However, in the case of Mycobacterium tuberculosis, we have seen that the system is not exactly the same. So if you compare with the Pseudomonas aeruginosa on the left part of the slide, with the Mycobacterium tuberculosis on the right part, so you see that there is an inclination of the RIP-C protein, that is the Lytic protein in that case, and the ATP is providing some increase in the angle of the RIP-C protein, how is compared with the membrane, and the release of the Lytic domain that was not visible in the cryo-EM maps. So with all these structures, we can provide a mechanism of Lytic regulation by FTS6. First of all, we have the FTS6 that is preformed, but with the extracellular domains that are extremely mobile. So MC can be then recruited but in that case, the Lytic domain remains occluded. And by the presence of ATP, that there is the trigger, that ATP triggers the elongation of the coiled coil, making the exposition of the Lytic domain. And at that time, so the amidases can bind the Lytic domain and then become active. So you can see this. This is the more or less the present state of the structures in different systems that more or less are providing some indications. While in the case of strep pneumoniae, there remain many questions to be solved. And another pending question is understanding how the Lytic system provided by FTSC, FTS6 coordinates with the biosynthetic machinery, the cell grid. So this is a remaining question. The final part of my talk concerns the regulation of serine-threonine kinases. Some years ago, the group of Christophe Grangeas discovered that the pasta4 from serine-threonine kinase interacts with the Lytic protein Lyt-B at the division septum. So it was really a surprising result because Lyt-B is a Lytic protein that somehow seems to be regulated by the serine-threonine kinase STKP. Lyt-B is a Lytic choline binding protein that is attached to the teichoic acid on pneumococcal surface. So the choline binding proteins are the most important family of surface proteins in strep pneumoniae. There are Lytic and non-Lytic non-enzymatic CBPs, all of them presenting catalytic domains and different repeats, so-called choline binding repeats. Thus confirm a choline binding module that is able to recognize the teichoic acids by interaction with the choline moieties. During these years, many structures of these choline binding proteins have been solved. some of many of them by our group. And in most of cases, we see that all of them present these elongated choline binding domains here in yellow, in which the choline moieties are represented here as spheres, are recognized at the choline binding site. The typical choline binding site is provided by 3 aromatic residues that perform some kind of cation P interactions in order to stabilize the choline moieties of the teichoic acid. So the regulation of the autolysins, the choline binding proteins, for instance, in the case of Lytic in pneumococcal fratricide, is produced mainly by modification of the peptidoglycan, as explained in this movie. So we are representing here the strep pneumonia, the pneumococcus, and pneumococcus is able to kill non-competent siblings by using Lytic. We have solved the structure of Lytic here in blue, and the three-dimensional structure of this protein avoids the entrance of the glycan chains into the active site due to clashes with the peptide stems. Attached to the peptidoglycan framework, there are the teichoic acid, here represented in orange. And so the competent bacteria release a toxin called CBPD that is attached to the non-competent bacterial cell wall of the non-competent siblings. So this CBPD presents peptidase activity, so it's able to break down the peptide stems in peptidoglycan. And so around the Lytic protein that is blocked by this peptide stem, so it's cleaving the peptide stems, providing the activation of the protein. So at that time, now the glycan chains are provided deprived of the peptide stem, so the glycan chains can enter into the active site, and so this protein that presents a lysosome activity can cleave the glycan chains very quickly. So the combined action of CBPD and Lytic release or make some kind of holes in the peptidoglycan framework. And so, as explained before, so this framework is essential in order to preserve the high pressure that is inside the bacteria. So, creating some kind of holes that can, the membrane cannot resist the pressure and explode. This is something that enhances the inflammatory process during infection, and also importantly, that the releases of the bacteria release also the DNA that can be incorporated by the competent siblings, and then providing increasing of antibiotic resistance. So this mechanism of pneumococcal fratricide is more or less well known. But the point is, as you have seen there, so the point is that Lytic requires the modification of peptidoglycan by other peptidases. But how can a kinase regulate Lytic? This is the question. So indeed, Lytic is a long story behind. So in 1967, so there were the initial studies by Alexander Thomas, who discovered that indeed modification of the choline by ethanolamine produced this kind of phenotypic effect, some kind of change in the bacteria. Then he discovered that this was related with the composition of teichoic acid, and only 20 years ago, so it was discovered that Lytic protein was responsible for this chain formation. So we have decided to follow a massive structural approach to unveil the three-dimensional structure of LitB. So we have done different constructs. No way for having good resolution for the full length structure, but very good resolution for the different fragments. So we have seen that indeed the catalytic domain presents two functional states, an open and closed conformation. We have discovered this mechanism, and then with the use of different peptidoglycan fragments, so we have seen the open state, the intermediate state, and finally the catalytic complex in which the peptidoglycan is fully recognized by the enzyme. And we have seen what are the residues that are essential for the catalytic activity and the peptide stems that could be recognized while the cross-linked neuropeptides are favored. But the most surprising result came from the analysis of the, in principle, boring choline binding module. So we have seen this very long choline binding module that present the choline binding site. You can see the choline molecules as represented as the spheres in this movie. And we have seen that besides the canonical site that is composed by these typical 3 aromatic residues, we have discovered new choline binding sites that are composed by 5 aromatic residues, 4 aromatic residues, or even 1 or 2 aromatic residues. So in the case of the sites we call hinge sites, this is impossible for choline to be recognized by these sites. So indeed, when we analyze the full choline binding module, so we see that there are different choline binding sites. The starting sites are located at the beginning of each domain. The hinge sites connect the 3 domains, and finally the C domain is presenting the most of the choline binding sites. So the question was at that time, why this choline binding module is divided in 3 different domains? What is the role of each domain? So by using Bacterial in collaboration, in very nice collaboration with the group of Christophe Janseas and Grangeas in Lyon, in France. So as you can see here in Streptococcus pneumoniae without LitB, so we observe this chaining phenotype. And if we make different construct, we add from outside the catalytic domain of the NM and catalytic domain, we observe always change. However, if we put the C domain together with the catalytic domain, at that time we observe the normal phenotype for LitB. So the conclusion is clear. C domain is essential for LitB activity, while NM domains are not required for LitB activity. So what could be the role of NM domains? So indeed, we have discovered that NM domains are required for cell localization. So if we make this GFP construction in which we are adding the full length protein and in a knockout LitB strain, so you can see how LitB localized at the pulse and at the septum. If we remove the pasta 4, inserting threonine kinase, we observe a delocalization of the protein. And if we remove an M domain, so we see also a strong delocalization of LitB. So the conclusion is that NM domains are required for LitB localization through interaction with STKT pasta 4. So we have performed molecular dynamic simulation to understand how the protein is recognizing the teichoic acid. So this is the structures we have provided molecular dynamic simulations. And we have also performed we have repeated by in silico the approach by Alexander Thomas 50 years ago by replacing the choline by ethanolamine and the interaction with the choline binding module. So as you can see here in the video, so the choline remains anchored to the choline binding module, while if we put ethanolamine here on the right, so you see that immediately ethanolamine is not able to remain anchored to the protein, so explaining the release of the protein and the chain formation in this phenotype. So interestingly, we have seen that there is recognition between the end domain and the pasta 4 of serine threonine kinase. So we have identified the residues that are making the interaction between the pasta domain in green and the end domain in yellow. So, and we have seen that the residues previously identified by the Grangeas group were essential for the interaction with DB, but also we have seen the essential residues in the end domain that are responsible for this interaction. So together, so we have a model of regulation and activation of DB mediated by serine threonine kinase. So in the first state, so there is a strong interaction between lipoteichoic acid with part of the choline binding module of DB, and then the pasta 4 is approaching the cell wall binding domain, and then establishing this strong interaction in the second part between the pasta 4 and end domain, and approaching this C domain to the wall teichoic acid that are located on the peptidoglycan framework, providing the right localization and starting the activity of the LID-B in order to cleave the peptidoglycan that is in between the daughter cells and providing the separation of the daughter cells. That's all. So as you can imagine, and also as explained by David, so this kind of studies is performed in collaboration with many groups worldwide. The group of Shahriar Mobashery and Mei-Lan Chan, University of Notre Dame, Christophe Grangeas, University of Lyon, Tom Bernhardt, Harvard Medical School, Martin Tan Bincher at Marburg, and Chris, Sam, and Luo Ming, Singapore, and Daniel Strom, and Sigve Hoverstein, and Norway, and our collaborators in Spain, Federico Gago and Ines Munoz. And the funding, I need to indicate that very recently we have started a Marie Curie action that is focused on the discovery of new inhibitors based on bacterial cell walls. So the idea is to use again the bacterial cell wall as a main target for designing of new kind of antibiotics. This is my group in Madrid downtown, and I'm happy to take a question. Thank you very much for your attention.
Thank you, Juan. There are a number of questions, and I will read them. Thomas is asking, do you have any favorite mechanistic model for how the divisome can synthesize cell wall against the turgor pressure of the cell membrane? How is the force generated?
Yeah, so indeed, this is really a very, very important question, and there is no clear answer for this. So because in that case, we have a coordination between biosynthetic and lytic machinery. So indeed, so we know that the Z ring is provided by GTP that is providing the energy in order to recruit different proteins in divisome. But also, as explained in my talk, ATP is also essential in order to really provide the energy for the activation of the lytic machineries. So the coordination between both systems is not clear right now. So this is the frontier of the discovery right now.
Okay. Jameson asks, do glycosidase autolysins like glucosamidase and muramidase and lytic transglycosylases also interact with protein complexes like FTS-EX, or is this interaction limited to peptidoglycan peptidases like amidases and endopeptidases?
With the present knowledge, there is no interaction between FTS-X system and other enzymes that are not the amidases in the case of gram-negative bacteria, while in the case of gram-positive bacteria, there is a direct interaction with a lytic protein. In that case, this lytic protein presents a coiled coil similar to the MC protein, but together with this coiled coil, there is a CHAP domain that is a peptidase domain. So, but there is different recognition of the other muramidases or lytic transglycosylases. To the best of my knowledge, there is no any specific interaction between the FTS-X system and any kind of lytic transglycosylase.
Okay.
Perhaps I could be permitted to ask a question.
Please, go ahead.
Juan, like everything else in life, timing is everything. And with respect to the FDS EX and AMI-AB story that you talked about, the release of these AMI enzymes could be catastrophic to the cell if they're released too early. So can you comment about how these complexes regulate that timing? How is that achieved? Why is that important?
Yeah, so you are absolutely right, Dave. So the point is that amidases in principle are intrinsically inactive. So only in the presence of these lead M domains that are acting as a hub in order to really trap the amidase. So only at that time, so the lead M domain is able to release this helix that is blocking the active site of the amidase and then provide the active conformation. If the interaction between the amidase with lead disappears, so at that time immediately the amidase becomes inactive. So how the full system is regulated is not clear. We are now starting, and you are also, David, doing a really very nice work on this kind of regulation as well. So how all this system is regulated in order to have ATP at specific moment in order to really release the catalytic domain and provide the activity, and how this is regulated and coordinated with biosynthetic machineries. I think is the holy grail right now in this system.
And if I maybe mentioned a second question on this, the last part of your talk about the LitB STKP system. So this is to me is a fantastic structural story. We never thought that these SD kinases with their pasta domains, that the pasta domains would be binding to other proteins. So this interaction that you've seen in this particular system, do you think we're on the verge of discovery of other types of interactions like this with the pasta domains of other proteins that contain the domain?
Yeah, so you know that it has been reported that the pasta domains in principle are expected to recognize peptidoglycan and somehow making the dimerization of the serine kinase. So as you mentioned, so this kind of regulation by the serine kinase is essential in cell divisions. Serine kinase is essential in cell division, while the mechanism is not really understood. And the pasta domains very likely is playing an essential role in this. So we have seen in the case of strep pneumonia that this pasta 4, but only the pasta 4, is making the interaction with the lead B protein, not the other pasta domains, among the 4 pasta domains. And also there is another interesting point in your question is that pasta domains are also present in PVPs, as you know very well, that only those PVPs that are involved in cell division. So very likely pasta domains are playing a very main role in cell division, but how they regulate the interaction with peptidoglycan or with proteins, this is something that we need to really uncover.
So it may not be just pasta domain interacting with the peptidoglycan, it may be pasta domain interacting with other proteins.
Yeah.
Perhaps we should stop at this point so we don't run too late, but please send in the questions and we'll come back to them at the end, time permitting. So the last speaker is myself. So let's see. Are we good?
You're good.
All right, thank you. So I shared this slide earlier. Cell wall is a single cross-link molecule that is indispensable to bacteria. We have had wonderful talks that expounded different aspects of the cell wall, which is wonderful as it makes my job easier. So What is known in terms of cell wall structure is the covalent interactions. So we heard about this NAG-NAM alternating structure, the peptide stem, the nature of the peptide stem, often a pentapeptide that has these D-alanine residues, bacterial. There's D-glutamic acid, another bacterial feature. The side chain makes the amide bond, not the main chain carbonyl. And then you have amino acids like diaminophenolate that are entirely bacterial. So this is a uniquely bacterial structure. Then, when two strands of peptidoglycan get to close proximity of each other, they have the opportunity to cross-link. And here, what you're seeing is the so-called 4-3 cross-link. And here is the 3-3 cross-link. So the cross-linking, it turns out, is absolutely critical. If you interfere with the cross-linking, bacteria die. So I just wanted to acknowledge the very important contribution by a series of talented synthetic chemists in my group that made possible many of the studies that I'm going to be talking to you. I won't have the time to describe the synthesis because of the shortness of the presentation. But Kazuyuki Miyashita was one of my first postdocs way back when, and he would go on and say things like this. Any compound with 4 bonds to carbon, 3 bonds to nitrogen, 2 bonds to oxygen can be made these days. He was a full-time postdoc and a part-time philosopher, as my statement says there. So why Kazuyuki said that was his confidence in his ability and the field in terms of making molecules of interest. In our case, biologically interesting molecules that are involved in bacterial physiology. Again, as a way of reminder, I want to say that beta-lactam antibiotic target penicillin binding protein, which David addressed very nicely. So these are bound to the surface of the plasma membrane, and they do their stuff on the cell wall. So beta-lactams, as in penicillin, have the ability to inhibit the transpeptidase domain of penicillin binding proteins. So when this inhibition takes place, the cross-linking event that is critical for survival of bacteria would not take place, and the organism dies. But when there is damage to the cell wall without organism dying, let's say sub-MIC concentration of the antibacterial, the transglycosylase domain, again, David gave a detailed presentation on the function of this protein, continues its polymerization of lipid 2, so it gets polymerized. However, because the transglycosylase is out of commission, it doesn't have the ability to be incorporated into the leading edge of the bacterial cell wall. So the reaction is shown down here, that you take the 2 sugar units of lipid 2 and make it 4 sugar, and so on, as David beautifully showed. So when aberrant peptidoglycan is generated, there are a host of enzymes referred to as lytic transglycosylases that come and fragment it. Their function, in a way, in this instance, is to remove the aberrant structure, the damaged peptidoglycan. But they have also functions during the growth of the bacterium. It's not necessarily in response to antibacterials per se, but they presumably have roles in excavating inside the cell wall for insertion of the pili and the flagella and the like. But regardless, lytic transglycosylases degrade the aberrant peptidoglycan in the case of exposure to antibiotics that damage the peptidoglycan. They generate a series of compounds, as we'll see shortly, the end product of which is a disaccharide with the structure of 1,6 anhydromuramic acid being the hallmark of the reaction of the lytic transglycoside. So these so-called muropeptides are brought into the cytoplasm by the function of AMPG, and they undergo a series of a dozen chemical transformations leading to lipid 2 formation, flipping the lipid 2 to the surface of the membrane, and resynthesis of cell wall. Off the biosynthetic pathway inside the cytoplasm, the molecules serve as signaling entities in regulating expression of genes. So one of the genes that gets expressed is the so-called AMPC gene that produces the class C beta-lactamase. What is beta-lactamase? Beta-lactamase is a resistance enzyme to beta-lactam antibiotics. So the beta-lactam came in, inhibited the transpeptidase. There was damage to the cell wall. Lytic transglycosylase fragmented the cell wall. Fragments came in and actually induced formation of the resistance enzyme that is exported to the periplasmic space to take care of the drug that is hurting the bacterium. Very elaborate, very interesting system that has evolved as an offshoot of biosynthetic processes. So Mi-Jun Lee in my group a few years ago set out to investigate what neuropeptides can be identified in bacteria. So Pseudomonas aeruginosa is one of the organisms of interest in my group, and the work is detailed and we published it, but the essence of it is that she went painstakingly, identified after you grow bacteria, you break open bacteria and go look for these neuropeptides. And not only by LC, she matched them to known samples that she and Dusan Hessek synthesized. These are authentic synthetic samples. 6 of them were mixed in this case. So she would go and by LCC compound 4D, matches it with one among the standards, and then so on. So compound 2D matches it with authentic sample, then the matching by mass spectrometry to make sure that all is well. So what she did, in the absence of antibiotic, she identified 20 different compounds and quantified them. So you see here, it says molecules per bacterium times 10 to the 4. You bear in mind that Avogadro's number is 10 to the 23 molecules per mole. So these are vanishingly small quantities of these samples that were identified by mass spec. So when you go ahead and expose Pseudomonas to sub-MIC of a beta-lactam antibiotic, 2 of these agents light up because their concentrations increase. And these are indeed signaling molecules in gram-negative bacteria for induction of resistance mechanisms. Mi-Jun went on to do the experiments in vitro as well. So if you isolate bacterial cell wall, you expose them to the individual enzymes. There are 11 lytic transglycosylases in Pseudomonas aeruginosa. So every one of those, the genes were cloned and the proteins were purified to homogeneity. And then you fragment the cell wall and identify the components that you identified. So here, cell wall is that sliver of green thing that encases the bacterium, and you take the grown bacterium, toss it into SDS, and boil it for a while, and then get rid of SDS at the end. So you end up getting the cell wall, the so-called sacculus, as a ghost of the bacterium, as you're seeing here. And then one by one, Mi-Jun went ahead and fragmented, and you get molecules like this. So here you're seeing a disaccharide with 1,6-anhydromuramic acid, the hallmark of the reaction of lytic transglycosylase. And in this case, there are 2 strands of peptidoglycan that are adjacent to each other, linked through a 4-3 bond. Here, two strands neighboring each other, linked by 3-3 bond, and then there are other permutations. So because we had these synthetic samples, you could do interesting experiments. So for instance, here are substrates to potentially lytic transglycosylase, and these are the products. These samples were synthesized for that authentication portion of the talk that I mentioned to you. So for instance, if you take compound 6 and do LC, it looks like this. You give it MLTA, one of the lytic transglycosylases, nothing happens. What that means is that compound 6 is too short for it being a substrate for MLTA. However, if you give it to MLTB, it fragments it into two products. Products 7 and 8 that you're seeing up here. And then she matched them with authentic samples by LC and by mass spec. So by so doing, for every 11 of the enzymes that I described to you, she went on to identify a total of 31 products during the log phase, and various enzymes have different activities like endolytic activity, exolytic endolytic activity being cuts in the middle of the peptidoglycan, exolytic from one end, a processive process. There are variations of their preferences. Certain enzymes prefer having this 1,6-anhydromuramic acid at the end, which is the product of the reaction of a certain other lytic transglycosylase. Others like the presence of peptide stems, yet there are lytic transglycosylases that do not have peptide stems preference. So every one of these are being studied. Many of them have been studied with Juan Hermoso for X-ray crystallography. These are the structures that you're seeing here. The simplest structure up to the more complex structure. Here they are, all of them, because I know that Protein Society members are very much interested in seeing the details. So normally in person, I get a big laughter out of this, but obviously we can't have that in webinars. But regardless, you see here a very large product for this system where you have 3 strands of peptidoglycan near each other, and they're all linked through the crosslinks. So we generate the aberrant peptidoglycan on exposure to sub-MIC of the beta-lactam because of the inhibition of the transpeptidase. Here comes one of the lytic transglycosylases, SLT, and I'm giving away the final message. This enzyme is involved in repair of the cell wall, and it comes and it indeed has both endolytic and exolytic activities. What it does is clips the backbone of the aberrant structure once in the endolytic reaction, and then a second time by processive 2-residue excision from the end by the exolytic reaction. So SLT is one of the functions of SLT is repair of damaged peptidoglycan. So we send the protein to Spain, to Juan's lab, and Juan came back with the structure, this beautiful donut-shaped structure. And then we had sent them also the tetrasaccharide substrate, which has peptide stems and so on. So he comes back and says that we got the structure, but somehow there are 2 substrates bound in the active site. So with this structure in hand, we notice that the space here can be filled with additional sugar residues to complete the structure of a bent peptidoglycan. Active site starts here and ends here. And to prove this, we supplied Juan the octosaccharide. Now, again, in the spirit of not minimizing the synthetic work, Dushan Hesek and Mijun Lee worked on this octosaccharide for about 18 months off and on before it was in hand. So what you see is that it spans the active site. There's this acute turn within the structure. It's an almost 90-degree turn. And there are surfaces for recognition of the peptides, because the full peptide exists in nascent peptidoglycan, which is off of the polymerase, the transglycosylase, that generates the peptidoglycan at the beginning. So here, what you have is, to the left is the arrangement for the endolytic activity. Here is the exolytic activity, which processive removal of 2 sugars at a time. And finally, the product complex that Juan beautifully developed. So here's a movie that shows the shape of this enzyme. The active site is in this region. It goes like that. And actually, substrate binding reshapes the conformation of the active site to accommodate the substrate. You see the 2 tetrasaccharides bound there. And here's the octosaccharide, the substrate for the endolytic reaction that takes place right around there. And this is the substrate that binds for the exolytic process, 2-sugar excision from the end. And finally, the product complex. So you have a sense of the full catalytic set of events for this lytic transglycosylase. So if lytic transglycosylase SLT is involved in repair of the cell wall structure, then what if we could inhibit the function of lytic transglycosylase SLT? If we could inhibit the SLT function, would that hurt bacteria because they can't repair themselves? So with that in mind, we went and looked at the literature, and indeed, there is a known, well, actually, there are 3 related inhibitors. They're referred to as bulgesins. And Takeda Pharmaceutical in Japan discovered them in the late 1980s. And we were interested in those. Two people, Dan Maroos and Mark Horstman in my group, worked on purifying the natural products from organisms that we acquired through ATCC, thinking that they should be producers. And indeed, by mass spec, we showed that bulgesine A was being produced. Unfortunately, we could never make it in sufficient quantity to actually see the compound in a test tube at the end. But if you go and do experiments with the extract, this is what we saw. If you grow Pseudomonas, in the absence of any compound, it grows just fine. In the presence sub-MIC of S3NM, which is a beta-lactam, it grows just fine. If you give the extract, it grows just fine. But if you give the extract and S3NM, bacteria die. And this is the growth curve for this process. Mark went on to do whole genome sequencing, and very interesting observation that he made was that the cluster for beta-lactam biosynthesis, in this case sulfazacin, this compound, is adjacent to the bulgesine biosynthetic machinery. In all likelihood, this organism prepares the 2 natural products simultaneously, and they're regulated simultaneously. Shusuke Tomoshige in my group went on and synthesized the bulgesins, A, B, and C. So I'm not going to talk about chemistry. but availability of the compounds allowed us to compare them, and they're roughly as good. And here's the bound structure to SLT from Juan's lab of bulgesine A. So these are experiments by David Dick in my group. If you take Pseudomonas aeruginosa that produces green fluorescence protein constitutively, bacteria look like this. If you treat them, with ceftazidime, which is a beta-lactam at sub-MIC, that's how they look. But the moment you add bulgesine A, you start getting these bulges at the point where septation would take place. So as bacteria are growing, they need to have septum for the daughter cells to separate. Instead, we are getting bulges. So what we have here is that the transpeptidase is being inhibited by beta-lactam, and SLT, which is the repair enzyme, is inhibited by bulgesine A. And David's electron microscopy picture shows these bulges. There's quite a bit of variety, stages of growth, and so on. And these two we interpret as the bulges have had their breach at this point, and the bacteria have died. So this is an experiment by David Dick. So he put in the middle the bacterium, Pseudomonas in this case, for it to grow outward. And on the red dot, one puts compounds. So here, there is, let's see, there's nothing here. And bacteria grow just fine, 14 hours and 22 hours. Then in the presence of bulgesine A, bacteria grow just fine. In the presence sub-MIC of ceftazidime, bacteria are filamentous because they cannot form the septum. It's sub-MIC. You get some death. That's the red color. The red color forms by reaction of the DNA that has been released into the medium, which has propydiamide. Propydiamide that complexes with the DNA to give the red color. But when you have ceftazidime and bulgesine A mixed, you start seeing death earlier. And of course, by 22 hours, the plate is awash in red, which means that most of them have died. So here is a real-time movie that shows this process. So please pay attention to these bacteria right here. Bulges have formed, bulges explode. It repeats itself. Notice that the green color disappears first because the organism stops making the green fluorescence protein, and then there is the breach of the bulge to signal the death of the bacteria. And here's a snapshot of the process. You see how many of the bulges there are. So this is an experiment that David did that actually is very nice. So you put the compounds ceftazidime and bulgesine A, you get your bulges. David went and removed some of that, put under microscope. Yes, the bulges are there. Then he put them on growth medium in the control group, which has no compound in there, so the bacteria should grow unhindered. And then in a plate that had bulgesine A present. And here are his results. When there are no compounds, the bacteria recovered. The bulges disappeared by and large, and the bacteria are thriving. If you put it on the plate with bulgesine A, the bulges persist, and bacteria begin to die. So bulgesine is interfering with the ability to grow and recover. Another experiment by David Dick. This is done 11 times with all the lytic transglycosylates. So what he started with was transposon insertion mutants for individual genes. So here's SLT has been inactivated by transposon insertion. This is akin to having an inhibitor that is selective for SLT. So the SLT function is knocked out, and then you see the consequence on exposure to sub-MIC of the beta-lactam. And what you see is the formation of bulges. SLT, MLTD, and MLTG have this ability. So in other words, the activity of any one of them is sufficient for fixing the damage. If we were to inhibit the function of any one of those three, the organism is in jeopardy of dying. Then David went on by mass spec to get dissociation constants for the individual enzymes, SLT and MLTD. Both of them are important as targets for lytic transglycosylate, and these are single-digit micromolar dissociation constants for those enzymes. I want to end my presentation by saying that what I have described to you today is, of course, the function of the individual lytic transglycosylates and their importance for various things that I've talked to you about. Here, we have a painting by Georges Shahriar on pointillism. Pointillism is a way of painting by putting dabs of paint on a surface that produces an image that is pleasant to the eye. But it's the collection of the points that you have added that makes the difference in this sort of painting. But in reality, the individual pixels are not that important, but rather the collection of the pixels. And what you're seeing here is his famous painting, La Grande Jatte. That is a beautiful painting, a collection of dots of paint on the surface. This is a painting at the Art Institute of Chicago. And the point is that I talked to you about the individual function of lytic transglycosylate or penicillin binding protein and whatnot. In reality, as we have seen in the previous two talks, is that there are whole machineries of enzymes that support the processes that are relevant for cell wall. So here as well, lytic transglycosylases are involved in other proteins in large assemblies that are critical for survival of bacteria. And here is an image that, divisome and elongasome that we have heard that are multi-protein complexes. But you notice in none of these images and others in the literature, there is a single lytic transglycosylase that has been invoked. And however, I want to point out the work of Luis Avila Cobian in my group, who did a series of experiments in pulling down the various things within the membrane and within the soluble portion that may attach themselves to various lytic transglycosylases. Especially this first paper, we showed all partners for all lytic transglycosylases of Staph aureus. And there are 71, I believe, proteins that partner up with lytic transglycosylase. And here is the story of RLPA and roughly about 25 partners that RLPA has. What came out of the pull-down strategy was verified in solution for association of the lytic transglycosylase with the individual one. So Luis went and cloned all these proteins. There was one false positive. And certainly, there's no association that we could see, but one wonders whether membrane environment may be necessary for that association. So he characterized these interactions by various biophysical methods. I think that brings me to the end of my talk, but this represents a frontier of understanding how these assemblies of proteins do indeed what they do, and what they do to each other in terms of regulation of function that the previous speakers addressed. So with that, I will end my talk with a movie as well, which summarizes many of these processes. So here you have Pseudomonas. We have lipid 2 being polymerized by the transglycosylase. Up here, the transpeptidase is cross-linking with the release of the terminal dealanine. So the cell wall is being assembled, but there are other proteins like AMPDH3 that removes the peptide stems. These peptide stems have a tendency to bind to other things like MLTF, which is another lytic transglycosylase of Pseudomonas that I didn't talk about. It undergoes a dramatic conformational change to cleave the peptidoglycan, fragments the muropeptides entered through AMPG, and they're processed by the enzymes of recycling. The end product of this, 11, 12 enzymes lead to the formation of lipid 2. Flippase puts that to the surface, and the cycle has been completed. What if the transpeptidase is inhibited by the beta-lactam? Cross-linking no longer can take place. You generate the aberrant peptidoglycan, which the organism identifies as aberrant. Here comes SLT as one of the 3 enzymes that repairs: endolytic activity, processive exolytic activity. Muropeptides are being released. These are the ones that Mi-Jun Lee identified. And what if we inhibit SLT? The repair cannot take place. Aberrant peptidoglycan accumulates.
Excuse me.
The bulges form. And that leads to the demise of the bacteria. With this, I come to the end of my talk. My group, during our summer group meeting, part of our campus, please come and visit. It is a tourist destination in northern Indiana. We are very proud of it, a good place to come and visit us and the campus. I will stop here, and if there's opportunity to answer a few questions, I would love it.
Good.
I think we do have time. There are a number of questions coming up in the chat.
I have a question, Shahriar. So
Yes.
you mentioned a bit, that at least in the case of Pseudomonas aeruginosa, but also in many other gram-negative bacteria, there are so many lytic transglycosylases that in principle are doing exactly the same kind of reaction.
Yes.
Can you speculate about why we need this huge number of lytic transglycosylases in a single bacteria?
Yes, thank you. This is actually an excellent question in the sense that I'm going to give a long answer because it's so important. If you eliminate any one of these lytic transglycosylases, or if you eliminate by knocking out the genes for multiples of these, the organism grows as if nothing has happened. So one reaction to this is that these enzymes are not important because the bacterium grows just fine. Another way of looking at it is that these enzymes are absolutely critical. They're so critical that their functions have built-in redundancy. So if you lose one set of hydraulics in your jet, there are two other ones that the jet can fly on. And that's the same principle. And we have verified this by actually doing the reactions of all 11 of pseudomonal lytic transglycosylates. And the answer is there's absolutely overlapping activity among these. So for example, endolytic activity is seen in at least 4 of them. So endolytic activity is critical for that damage control that I just described to you. And 3 of those are known to play a role. So in our opinion, lytic transglycosylases have different functions. They have different partners. I didn't get a chance to talk about that. So the work of Luis shows that they interact with a whole bunch of different periplasmic proteins. They have different functions, yet there are sufficient overlap their activity that the loss of function of any one of them does not seem to be a total disaster.
Fantastic talk, Shahriar. You beautifully put together chemistry, biology, and the physics, and the biophysics in a wonderful way. And I clearly need to get some better movies. That one at the end was totally... I love the connection you made between the beta-lactam ceftazimidine and the SLT. And they seem to be working synergistically, even at an MIC for the beta-lactam, which, if my memory serves, is PBP3 specific, and therefore cell division specific. So given that synergy, do you know of other compounds which might inhibit LTs involved in the elongation process and work synergistically with compounds like methicillin?
Yeah, so that's a very multifaceted issue that one has to address. I want to say that in general, beta-lactams that target septum formation appear to be more critical than the ones that are involved in elongation. And the aberrant beta-lactam is going to form regardless of whether you're inhibiting the elongation part of the growth of peptidoglycan or septation. But whether there is a clear distinction between the two in terms of the repair mechanism, I don't know the answer to that. Suffice it to say, in Pseudomonas, and we picked Pseudomonas for 2 reasons. Pseudomonas, number one, is a very tough organism to address clinically. So we wanted to know more about Pseudomonas. But also Pseudomonas has one of the most number of lytic transglycosylases that there is. There are typically 4 to 8 lytic transglycosylases per organism. Pseudomonas has 11 of them. And we were curious why that is. 3 of them are clearly involved in repair of damage to the cell wall. Which one is going to be triggered at what stage, we don't know. SLT and MLTD localized to the septum. That's for sure. But how the organism engages the damage to the middle part of the peptidoglycan, I don't know present.
Okay. And I guess the reason, excuse me, that most clinically used beta-lactams that we know about are targeting PVP3 is because at that moment of cell division, then the cell is at its most vulnerable stage, because you're both synthesizing the new peptide-like and you're cutting the old to allow cell separation. So it's a
To
vulnerable.
add to that vulnerability is this bulge formation, which appears at the point of septum.
Right. Okay.
I have a quick comment, maybe, but it's probably a long discussion. So these talks, I think, just amazingly illustrated so many details of a very, very complex problem if you're trying to think about antimicrobials and antimicrobial resistance. And I'm just wondering if we're drawing it out kind of broader, right?
Mhm.
If we're thinking about antimicrobial resistance, what do you think are the prospects? Do you want to make a comment on anything from what we've heard about?
As you described it well, it's almost overwhelming because there are so many different mechanisms of resistance. And you think that one set of mechanisms you have addressed, like beta-lactamase inhibitors that have been available clinically since 1985, and you think that that's a solved problem, yet other issues pop out. The thing is, nature is on the side of bacteria because they grow rapidly, they mutate, and there are opportunities for selection. There are a humbling range of mechanisms for resistance that are known, and there are a zillion that are not known. So those of us who work in this field will not run out of subjects for study.
But it seems to me there are also, there's so much more known. There's going to be additional targets. It's just, yeah, we're heading towards the close here. I want to just make sure if there's anything that anyone would like to add.
Just on that point about resistance, we often think about antimicrobial resistance generally as being mediated by mutation. Well, in the case of beta-lactams, mutational resistance is actually quite rare. There are only certain organisms in which mutational resistance actually seems to play a part in resistance to beta-lactams.
Yeah.
It's predominantly beta-lactamases, or the acquisition of beta-lactamase genes. In the case of Staphylococcus and methicillin-resistant Staphylococcus, MRSA, that's the acquisition of a PBP that can keep working even in the presence of most beta-lactams. And in some organisms, the way that they become resistant is to switch their cross-linking mechanism from the kind of cross-linking that we've been describing, which is between the peptide stems at positions 3 and 4 to positions 3 and 3. So this 3-3 cross-linking mechanism is predominant in quite a few organisms. TB, for example, E. coli even does this when it goes into stationary phase. And I recently discovered that C. diff seems to have 3-3 cross-linking as its predominant mode of cell wall biosynthesis. So there's many ways to go about this, but resistance, as it were, is inevitable. However, beta-lactams have been the mainstay of our antimicrobial warfare since the 1940s, and they're still good today.
Sure.
So it's not over yet. And my last comment.
There's another mechanism, David, that Juan mentioned briefly in organisms such as the pneumococci. They can actually pick up from the environment whole pieces of DNA and incorporate into their own. Mm-hmm. So the issue is the rapid growth of bacteria and their extraordinary ability to utilize their resources in a way that is advantageous to them.
Yeah. So I would add that it is important to really understand the molecular basis of antibiotic resistant mechanisms in order to really try to really render susceptible these bacteria. So we have provided, in collaboration with Shahriar recently, a very nice mechanism in order to render susceptible MRSA. but also I think it's important in order to understand the molecular basis in order to really find new targets in such a way that, for instance, cell division or other essential mechanisms in bacteria can be used as new target for designing specific inhibitors. So we will have always bacteria that are resistant for any kind, because bacteria have been surviving for millions of years. So we are not going to discover the fantastic super antibiotic in order to clip and to kill all bacteria. But the point is that we need to really try to reuse the previous antibiotics to render susceptible bacteria and to find new targets in order to really continue in the fight against this very urgent problem.
I think all these talks exemplify the fact that basic science is very necessary in order to have translational outcomes at the end of the day. We need to understand resistance, we need to understand new targets and new possibilities.
Yeah. Absolutely.
I'm seeing this in the chat too. Yes, I think I want to thank you all again for this really beautiful webinar with I feel optimistic knowing that we are learning all these things, that we will prevail. So I also want to just mention that the talks will also be available on YouTube. So that's for our audience to know. And thank you all so much again for sharing this with us.
You're very welcome.
Very welcome.
Thanks, Liz. Take care.
Thank you. Bye-bye.
Thank you.
Bye-bye, guys.
Bye.